Thank you. So it's my honor this evening to introduce our speaker. Tonight we have Dr. Carrie McDonough from CMU. She is an assistant professor there in the Department of Chemistry. Very brief bio on her background. She got her bachelor's from MIT. Her graduate work was done at the University of Rhode Island Graduate School of Oceanography. She's had kind of a wide variety of experiences. So I think we're going to be in a real treat tonight to hear about her work on PFAS. A lot of her research resides at the intersection of environmental analytical chemistry, environmental health engineering, and chemical biology. She's interested in understanding how organic contaminants impact aquatic ecosystems and human health. So with an emphasis on chronic exposures to complex environmentally relevant mixtures. And tonight she is going to be talking about PFAS and sifting through these forever chemicals using high resolution mass spectrometry. So please join me in welcoming Carrie.
Thanks so much for the introduction and thanks for having me. All right, got all my things where I need them. Okay. So yeah, I'm going to be talking today about my use of high-res mass spectrometry to monitor polyfluoroalkyl substances, PFASs, in the environment. And these are chemicals that are called forever chemicals because they basically don't degrade on geological timescales. So a little bit about me just as I get started. So I run a research laboratory for chemical biology and environmental analytical chemistry at CMU. And the kind of motivation behind our group is asking, we know that past chemical releases were problematic. We know that there's a toxic legacy that has been left by legacy organic pollutants. So we have to ask ourselves, what is it that we might be using or releasing at this very moment that could contribute to a future toxic legacy for our own children and our grandchildren. So that's kind of the motivating question of our work. And we also, want to help solve those problems. So how can we prevent release and how can we mitigate effects? So I work with high-res mass spec and environmental monitoring to kind of trace organic contaminants from the source through the fate, so in the environment, and then into the ways that we are exposed to these chemicals, leading to an internal dose. And then I also work on a lot of interdisciplinary collaborations with toxicologists and epidemiologists, as well as metabolomics experts, to understand effect, because that's a really complex question that we definitely can't do on our own. To understand not only what's the dose, but how does susceptibility then lead to health impacts and, the actual reasons that we're really concerned about a lot of these chemicals. So today I'm talking about per and polyfluoroalkyl substances, PFASs. And these are really useful compounds that have been used in a lot of things because they're really useful. So they're very stable, as I mentioned, and oleophobic. They have many, many uses. So these have been used widespread in lots of different consumer products, making Teflon, coatings for different foods and grease repellent kinds of materials, waterproof materials, as well as stain repellent materials. So sometimes carpets are treated with these, textiles are treated with these to reduce the risks of, getting things stained. They're also used widely in aqueous film forming foams, so AFFFs, that are used to fight Class B fuel fires. So when you have a fuel fire, you want to starve the fire of oxygen as quickly as possible. So these foams basically allow you to make a film over your fire and then kind of reduce its oxygen supply and fight that fire more effectively. So they're very important for those kinds of applications. So clearly, lots of uses and lots of ways that we could potentially be exposed to these compounds. So one of the interesting things for us about PFAS is that they are fairly soluble compared to most of the legacy organic contaminants that we usually think about, like PCBs or something like that. So they can kind of hijack the water cycle. They can move in the groundwater, they can move in our water infrastructure, they get into our drinking water, and they're not as effectively removed because they're not super absorptive. And a lot of the ways that we treat water are through absorptive technologies. So industry releases, again, the HFFs, these things end up in the environment, can get into the waste infrastructure and then be released through that waste infrastructure. We also have direct exposure from consumer products. So we can get some of this from the environment. We can also get some directly from things that we use and wear. And why do we care? We care because these compounds have been associated with several different health effects. So this is a review from Sue Fenton et al. 2020. The solid lines are things that were fairly well established, high certainty associations with health impacts, and then lower certainty in the dotted lines. And you can see one of the big challenges with PFAS is that there are a lot of effects that they're associated with, including a lot that are very difficult to trace and track, like cancer. Cancer is a very long-term condition that, it's very difficult to trace someone's exposure over time to figure out how that really contributed. It takes a long time to figure that out. So many adverse health effects, some of which have been established quite well. And then we also have to realize that most of these studies that were reviewed here and that have come out since are all about two compounds, PFOA and PFAS, which are the C8 PFASs. Very few mixture toxicity studies and very few studies about any of the thousands of other compounds that we're concerned with. So this is what PFAS looks like as an example of what one of these compounds looks like. Extremely persistent, aquatically mobile. One of the best studied of the PFASs is PFAS. So we see these strong carbon-fluorine bonds that really difficult to break down. Bulky hydrophobic fluorinated chain, so this tail, perfluorinated tail. And then we also have a polar head group. So at environmental or physiological pH, this is going to be an anion. We also have compounds that we call precursors. I also call them pre-PFAAs to be more specific sometimes, because precursors can mean like a billion different things to different chemists. But these are polyfluorinated compounds. So we have perfluoroalkyl acids, and then we also have these polyfluorinated precursors. And these are any number of different various structures that have some perfluorinated moiety, but then also some part that maybe is a hydrocarbon. So they can break down theoretically over time to form perfluorinated compounds. So their final product of something like this would be something like this, or with a perfluoroalkyl carboxylate. So that chain length is really important to the behavior of these compounds. I mentioned the bulky hydrophobic chain. So when we have a short chain, smaller tail there, we have a more soluble compound. That anionic character kind of comes into play more. It's more mobile in water. So you get something more soluble, less hydrophobic. And then as we go up in chain length here for the perfluoroalkyl sulfonates, we see more and more hydrophobicity, more bioaccumulation, and less solubility, so more absorptive. So a lot of their partitioning and behavior in the environment is governed to some extent by this chain length question. So getting into PFASs in our bodies and the accumulation in living things, this is a story that has fascinated me for a long time, because the story of understanding exposure really to anything is a story about the advancement of analytical chemistry. The more that we understand how to look for things and how to see things, the more we know, what we should be worried about. If we can't find it, we have no idea that we should be worried about it. So we actually knew pretty early on that PFASs were likely in human blood. So Donald Taves in 1968, analyzed his own blood, which I have never done and I refuse to do. But I guess he just took it in his own lab and he analyzed it by NMR and found that ashing made the fluoride more available. So there's more fluoride after burning, basically, than there was before, which is evidence of inorganic fluoride as well as an organofluorine component in human blood. So he said, gee, this is interesting and it's in me. It's consistent with the presence of a fluorocarbon. So then he went and looked at, I think, about 30 other participants. So they just got a cohort together and did some blood analysis and found that all of those samples were also contaminated with these organic fluorocompounds that they assumed were probably derived from commercial products. It's kind of as far as they could go with the technology that was available to them at the time, but they were already pretty close to uncovering this problem. So as things kind of continued, LC-MS became the gold standard of how we analyze these compounds. It was not this homemade LC-MS, but that technology enabled us to do some really nice targeted analysis of these compounds. So once we could make fragments, we could really see what was there. And this is a paper by Hansen et al., 2001, that was demonstrating some of the first use of LC-MS to analyze PFASs in biological materials. So they found an unspiked human serum sample, and they actually ended up using a rabbit serum as their blank because they found that they could not find analyte-free human serum. So they couldn't find a blank to use. No one didn't have this stuff in their blood. This was something that was a big struggle for a long time because no one believed that that was true, but it was actually just that we were so late to the party that there were no blanks left. So pretty scary to find that there's, no analyte-free blanks among human serum. We now know, using these LC-MS methods, that PFASs are, unsurprisingly then, present, they say, in over 99% of human beings. So pretty much anywhere you look, you're going to see these in human beings. Maybe they're a little below detection limits in a couple people. And they're particularly elevated in people who live in impacted populations. So areas that have been impacted by HRMS, those aqueous film forming foams, have higher levels of these in their drinking water. And there are several other sources like that as well. So drinking water is kind of a conduit for some of the higher exposures that we see, but they are ubiquitous in the environment. Everyone's being exposed to lower levels. So the highest levels that we see in people have been manufacturing workers and people in areas with highly impacted water. So by virtue of, these compounds being in everyone's blood, that means that fingerprints of these compounds are in everybody's blood. So we can look at that composition to start to learn about sources, how they interact with the body, how that external exposure relates to internal dose, which is what I'm really interested in. So I like using this slide because I like looking at chromatograms and I find it very informative, but just so you know, these are representative samples. We have a lot more samples that show the same average composition. So I just kind of picked, I would never, make blanket statements based on one compound's chromatogram. It's just a nice way to look at it. So this is human serum data that I analyzed from the World Trade Center Biobank at Stony Brook University. I think this was a sample from 2011. So just looking at, kind of the general signature in a first responder, someone who does a lot of this kind of work, probably has used foams, those kinds of things. And you can see our chromatogram here. So we're going from more soluble to less soluble. This is reverse phase LC. And we have a number of these perfluoroalkyl acids showing up. So we have PFOA, we have linear PFAS, and then we have branched PFAS, quite a high signature from that branched PFAS. So when we have electrochemically fluorinated PFASs, we always have this signature of a big branched group of different branched isomers and then the linear separate. So that's pretty signature to that. And then we have a few other compounds as well. And I like to show this because it really shows how different populations contamination can be. So this is a shark, a white shark from the Northwest Atlantic. So very different population, different physiology, different animal, clearly. Concentrations are much lower. So our total perfluoroalkyl acids in this shark were about a nanogram per mil plasma, whereas in humans, probably about like 5 to 8 nanograms per mil is pretty normal. 30 is quite elevated. And some things we can just start to learn just from looking at the composition of these things in blood. So here we see a lot of compounds coming out at these later retention times in our sharks, a lot of ultra long chain perfluoroalkyl carboxylates, which are a signature of biomagnification. So these are really, really long chain hydrophobic compounds that biomagnify in fish and then magnify up the food chain in these apex predators. We actually see a kind of similar signature in people who eat a lot of seafood, which I might have a slide somewhere in here about that, I'm not sure. So you can see this is a really different signature. You can also see that our sharks have almost no branched isomers of PFAS, whereas our humans, particularly first responders, but this is true of really anyone, have much higher levels of that branched signature. So that's kind of an interesting thing that we've been starting to try to tease apart, is why maybe direct exposure or closer exposure to these compounds might lead to higher levels of branched than what we see in wildlife in remote areas. So they're everywhere and the fingerprints are everywhere. Here's some more shark data just to drill down on that a little bit, because I published this paper pretty recently and I was really excited about it. I think it was our first research paper that I got to be corresponding author on. So I was pretty excited about that. So this is PFAS fingerprints in white sharks. We collected these samples from OSearch. So they go out and they tag sharks and then they take serum and small muscle samples from those sharks and then let them go. So we got about 30 samples of plasma as well as some muscle samples and looked for these PFASs in these sharks. And we kind of divided them by location. So you can see at the top, we have Nova Scotia, Massachusetts, Carolinas, and Florida. So these are captured off those coasts and each line is an individual shark. It's not that easy to catch sharks. So this is a pretty good number, sample size actually for sharks. But it is still a very small sample size. So we see again, those long chain PFCAs are quite abundant. They appear to be bioaccumulating and biomagnifying. And we also see some odd chain PFCAs. So PFCAs, perfluoroalkyl carboxylates, and their precursors are fluorotelomeres, or their precursors, are usually manufactured in even chain lengths. And when we see a pronounced odd chain signature, that usually means that we're seeing degradation of atmospheric fluorotelomeres in whatever receptor we're looking at. So probably indicating fluorotelomere alcohols or other volatile fluorotelomeres that are being deposited in ocean water and then either transforming before or after their accumulation in these animals. We also saw a lot of interesting geographical trends. Again, we have a very small sample size because sharks are pretty elusive, but some kind of interesting conclusions that we can start to draw. Mainly, we saw that for a wide range of compounds, concentrations were greater in Nova Scotia. So in the most northernmost area where we were able to collect samples, and then went down in the more southern areas. We think this may be because of diet, because these sharks migrate every year. They go down the coast and then they come back to the same place. So if you're a Nova Scotia shark, you'll migrate around, but you'll always go back to Nova Scotia. And you probably maybe eat more large marine mammals that also have bioaccumulated maybe more amounts of these compounds. So that was one theory that we have about this. We also have thought that maybe this could be due to differences in, physiology and metabolite rate when you live in a really, really cold environment versus a warmer environment. So those are things that we're kind of ongoing thinking about. But we do see a significant geographical trend here. So I showed you some fingerprints of PFASs and the kind of things we can learn from these targeted analyses. But then the next question is, what about other PFASs? There are thousands of these things. So I also like to pose in front of my mass spec. And as we get, more and more powerful instruments, we find more and more of these compounds. We know now there do seem to be thousands of these things. And we're now using this new Agilent 6560 ion mobility mass spec in my lab to kind of continue probing this question of what else is out there. So we have this whole PFAS universe that we want to explore. There's a lot of different analytical techniques to use for that. One of the most inclusive, I guess, that gets the most fluorine would be PIGGY, so that's particle-induced gamma ray emission. So as I go down in the sizes of the boxes, I'm talking about smaller fractions of total PFAS, basically here. F19 NMR is pretty good for getting a pretty complete total organofluorine signal, and you also can see some information about connectivity, which is valuable. People also use extractable organofluorine or adsorbable organofluorine. So these are methods kind of like what Taves used, where we're preparing a sample, kind of pulling out whatever sorbs and stays, and then we're burning it to see what kind of fluoride comes out. So it's not going to give you structural information, but it'll give you a total amount. And then we get into our mass spec methods. So LC HRMS is more commonly used. GC HRMS is becoming more and more used for GC amenable PFASs. We have TOP assay, which allows us to infer precursor content based on oxidizing samples. And then in our littlest squares, we have the things that we use the most. Really, LC-MS-MS is the thing that we use the most for targeted PFASs. You're going to get the highest confidence in what you're actually seeing and the best selectivity, but you're also going to miss quite a lot of the picture when you're using those methods. So a lot of studies use this kind of LC-MS-MS targeted PFAS, and then they'll also use something like F19 NMR or EOF, AOF, one of those larger integrative methods. And by doing that, you can compare your two methods and the total mass that you get out of both of those methods. And you can measure unidentified organofluorine. So now you not only know what are the targeted analytes that I'm able to measure in my sample, but you also know how much more organofluorine is in my sample that I cannot identify. So this is kind of, I would say, as far as we've gotten with probing the mass balance, is kind of getting to this point where at least we know to some extent how much we don't know. Didn't want to move on from there. Now it does. So we call this PFAS dark matter. This is the unidentified organofluorine fraction. This is from a review that we published recently in Trends in Environmental Analytical Chemistry, summarizing all of the studies that have measured unidentified organofluorine in biological samples from humans. And you can see the range is huge. We go from about 0 to 100%. So it doesn't really tell you much other than that there's enormous inter-individual variability. Some of that is probably due to pharmaceuticals. People who use fluorinated pharmaceuticals are probably going to have higher levels in their blood, and those are often not in our target lists. Some of the other interesting observations we had from this are that generally females tend to have higher levels of unidentified organofluorine than males. There's increasing fractions of unidentified organofluorine over time in samples. So it seems like the amount of this stuff we can explain is decreasing. And there's an inverse relationship with total organofluorine, which means if you're in a really contaminated area and you have quite a high body burden, often that body burden is mostly explained by, compounds that we can measure. If you're just in the general population living somewhere where maybe you're using consumer products and other diffuse sources, you're much more likely to have a lower total concentration in your body, but for those compounds to be unexplained or things that we can't identify as well. So because there's all these, thousands of compounds, we use high-res mass spec to look for PFASs, and that's what my group has been doing a lot. We like it because it, enables that better understanding of the full extent of contamination, but it still gives us structural information. So we're not just getting a total organofluorine, we're getting some information about what's actually present. It also helps to improve our differentiation between exogenous PFASs and endogenous compounds. So there's lots of bile acids and other things in biological samples that masquerade as PFASs that you cannot differentiate using a low-res mass spec. So quite a few regulatory organizations at this point have said, for PFBA and PFPEA, which are the very short chain perfluoroalkyl carboxylates, you have to use high-res mass spec if you're going to say they're in food or in biological samples, something like that, because the interferences are just too common. So that makes high-res mass spec very attractive for this kind of work. This is the workflow that we generally use in my lab. It's a suspect screening top-down non-target. So we're doing non-target. We could look for everything, but to save ourselves some time, usually we use an extracted ion chromatogram list that has, most of the things that we suspect that we might see. So we use a list of mass and formula for more than 1,000 characterized and theoretical PFASs, which we adapted from NIST. We look for exact mass matches within 5 to 10 ppm. We look for isotope pattern matches. And then for a subset of those compounds, probably about 300 of them, we have fragmentation library data, so we can get a more increased confidence in our identification. But there's, thousands of these things, so we don't have them all in our databases. So that's how we've been doing this. We use an Agilent. This is a 6545, but now we have a new instrument in our lab that also has ion mobility. So we have a drift tube as well on the kind of front of this same instrument, but it has one more box basically. And we're really excited about that because now that we have ion mobility, we can more rapidly separate out the kind of halogenated xenobiotic space by mass to charge versus size, so collision cross-section, versus endogenous compounds. So hydrocarbons are going to be much larger in size for the same mass as a perfluorinated compound. So you can see this is from Aaron Baker's work at UNC, which convinced me to get an ion mobility mass spec. And they have this really nice separation where you can just pop out those halogens, which is really nice when you're working with messy biological samples. So a demonstration of kind of how we use that ion mobility and how we're hoping to use it. It's pretty new. We just got it last summer when we moved to CMU. But we've done a little bit of work with it so far. So this is, again, an example of these PFAS fingerprints. And this is the one I was promising you before that I would depress you with. So this is our first responder again on top. And then we have an avid seafood consumer from Long Island on the bottom. And both have quite high levels of PFASs in their serum, but very different signatures, right? So we have this high branched signature in our first responder that isn't really there as much in our seafood consumer. And then we have those longer chain carboxylates showing up in our seafood consumer that you actually can't see in our first responder. So again, echoing that biomagnification from seafood. So we wanted to probe further into what are those branched isomers and what does that mean for, potentially source tracking and things like that. So we've been doing a lot of work on these branched isomers. This is a review we wrote on kind of their behavior in different environmental compartments. So you can see generally in the environment, we have higher levels of branched isomers. And then as you go up into organic carbon and bioaccumulation and living things, you see more of a signature from the linear, and the branched actually is more readily excreted. So we've been looking into this with our instrument. One of the reasons we wanted to look at it is because we know that these compounds all have different toxicokinetics. So all of these branched isomers are going to have different excretion rates from the body. And usually we just always quant them as just some branched. So we just do chromatography, you get like a lump out that's your branched, and then you have your linear. You can tell those apart, but the branched is just all the branched together. And this paper came out from Jochen Mueller's group, Nilsson et al., where they showed that actually those compounds all have, a different half-life. Every single one of those branched isomers behaves differently. So this is some early work that we've been doing, visualizing those branched isomers by drift time and retention time on our ion mobility mass spec. So you can see we can't quite pull all of them apart, but compared to just using chromatography, we are getting closer and more efficient with pulling apart what this composition is of these different branched perfluorooctane sulfonates. And here's the branched profiles for 3 individuals from that first responder cohort. So starting to tease apart what are the actual branched isomers in here. They look quite similar among this cohort, but then the next question is, what do they look like if we compare that to, seafood consumers or people who don't have any, specific source of exposure. So that's something that we're starting to look into now, which has been enabled by having this ion mobility capability. So now I'm going to move into talking a bit about how we're using these things overall. And we're doing this, basically we're doing a lot of dosing of biological systems, mostly in vitro, but we've done some in vivo with complex mixtures. So I'm going to be focusing on a study that I did with Jamie DeWitt, who was at East Carolina University at the time. She's at Oregon State now. She's an immunotoxicologist who does a lot of really great PFAS work, and she works with mice. So we dosed mice, C57 black 6 mice, for 10 days with an aqueous film forming foam. So the entire mixture of both known and unknown PFASs that are in this mixture. And then we let them depurate for 6 days and collected serum. We collected urine throughout the study at several different time points. So here you're seeing just the serum levels that we had via targeted analysis. So these are the compounds that we actually had analytical standards for. And as you would probably expect, the concentrations in serum go up as the dosing level goes up, which you would expect. But we wanted to use this model to start to ask some other questions. So I was really fascinated with the idea of using this kind of complex mixture where you don't know everything that's in it to prioritize novel PFASs that might be bioaccumulative. So is there a way that we can, by dosing an animal or using an in vitro technique, start to prioritize compounds that bioaccumulate more that maybe we didn't even see in the initial mixture? And then we also want to find out, are these kinds of dosing studies, do those help us to do human biomonitoring where the concentrations are lower and we have a really difficult time teasing things out? So we've been doing this in this kind of schematic. So we have HRMS and other complex mixtures, dosing them into biological systems, tracking transformation, by which I mean both biological transformation products and the difference or the transformation in composition. So some things are going to be accentuated by bioaccumulation, some things are not because of excretion. And then finding novel compounds, doing HRMS characterization, and finally using them to inform human biomonitoring. So informing our databases that we use to track human exposure. This is what the foam that I use a lot looks like. So this is a study we did with Jeremy Komel at Yale. He's working on making fluoromatch. So we did this with him to give him some HFLF data, basically, and are now working with him on doing some ion mobility as well. So you can see all these different homologous series are all different subclasses of PFASs. And then we have examples of some of the structures. So we have perfluoroalkyl acids, we have substitute perfluoroalkyl acids, and then we have all sorts of precursors that have different alkylated functional groups on them. All sorts of things, it's a really messy synthesis process, and we end up with this kind of sulfonamide perfluorinated chemistry. And we're concerned about this mostly because there was a lot of direct exposure to these kinds of foams. So not only are we exposed through drinking water and through them getting into our groundwater and kind of laying there for a long time and eventually getting into drinking water wells, also people who work with aqueous film forming foams in the Navy, in the Air Force, firefighters have had direct exposure to these. For a long time, it wasn't known that they were dangerous or that these compounds were toxic, so people really didn't even wear PPE. So we want to understand the consequences of being directly exposed to these foams. So this is the serum of one of these HFF dosed mice after deparation. And we're looking at a bubble plot. So this is high-res mass spec data, retention time, and the mass to charge ratio on the y-axis. And then the bubble size is kind of the relative peak area of the different compounds. And you can see all of these different substituted perfluoroalkyl acids showing up, including the ones that I've kind of pointed out here: unsaturated PFAS, oxygen substituted, and ketone substituted, which were pretty highly accentuated. And it suggests that these novel compounds, which really haven't been studied much, seem to also be bioaccumulating in our model. So we calculated kind of the relative enrichment of these compounds by normalizing them to PFAS, which we know is a bioaccumulative compound, and looking at kind of how does the composition in this mouse serum compare to what we dosed with. So is it, more accentuated than it was in the dosing solution or less so? And we had to do this kind of pseudo bioaccumulation factor because there are no analytical standards for most of these compounds. So we can't do traditional quantification. But what this suggests is that unsaturated, ketone substituted, and hydrogen substituted PFAS were all enriched in our serum samples to a greater extent than they were in the HFF and to a greater extent than PFAS was, suggesting that they're at least as bioaccumulative as that PFOS, which we know is bioaccumulative. A note on the fragmentation of these compounds. It can be really challenging to tell them apart. So here we had a compound that we saw in our mouse serum, looks pretty much the same as this analytical standard for PFAS, which is perfluoroethyl cyclohexane sulfonate. So we've got the same two fragments there when we look at it in 10 EV, and then we go up to 25 electron volts. So we're going up in power in our collision. And then once we go much higher, so we're up at 40 EV now, you can see they're actually distinct. So this is actually an unsaturated PFAS, not a cyclic PFAS. But you can't really tell unless you bombard these pretty hard. So PFAS don't like to fragment, and it takes a lot of power to fragment them well enough. So one of the other questions we had about this data was, usually, as I showed before, we expect these to get more hydrophobic and more bioaccumulative as chain length goes up. But here we saw that HXS and HPS were much more, highly accentuated than the other compounds. So it seems like there's some kind of perturbation of our chain length trends in this in vivo system. And we think that this is because of precursor transformation. So these sulfonamide-based precursors are shown here. This is the HFF we used in dosing, just spiked into serum, so not used in a dosing. And you can see all these gray circles are precursors, and then they're all gone when we look at post-deparation mouse serum. So either they were excreted or they were transformed to sulfonates. And that's kind of what we've been working on now, is this question of transformation. So I'm going to go to the parts I'm excited about. So this is my student David's work, who's at Stony Brook. He's probing kind of the biological fate of these sulfonamide-based precursors in these mice. So using the same samples that I collected in my postdoc, and we've got a ton of samples and non-targeted a ton of data, so this is going to take us forever. But he's now looking, how much of these precursors is accumulated, transformed, or excreted? Where are they actually going in the body? So he's been doing this work, doing a lot of analysis on mouse urine to look at, do we see precursors coming out of the animals over time when they're dosed with these HFFs? And he found this interesting series of, it looks like a PFAS series, but it's a higher mass and a lower retention time than the perfluoroalkyl sulfonamides, which are sulfonamide precursors. And usually we just throw things away when we're doing non-target screening and we see something that has, a much higher mass and a lower retention time. That's usually considered kind of nonsensical. But here we have a metabolic conjugate. So we added something, we got bigger, but we also got more soluble. So we see this consistent retention time decrease because we have glucurinated FASs here. So we had a series of different metabolic conjugates. Some of this has been seen in vitro before, but this is actually the first time I've seen it in vivo being excreted from a actual animal. So this seems really useful in terms of, if we want to do urine screening in humans, this is something that we might want to look for. Another thing I'm really excited about with this data, which we'll hopefully have published quite soon, is the branch signature. So here we're looking at the parent FHXSA, so that's the C6 sulfonamide coming out in urine from these mice. And we have a branch signature that looks pretty big at the beginning and then goes down, and the linear stays around longer and is being excreted for longer. And then we have this conjugate signature, which I am still puzzling over, but very excited to start looking at the ion mobility data for and see if we can learn more about what those structures are, because it seems like maybe we have more branched isomers being conjugated than we have being excreted as parent compounds. So my final little thought here that I wanted to give you was one of the reasons that motivates me wanting to do this kind of work where I'm looking at complex mixtures, but usually pretty simple biological systems. The mice were pretty complicated, but we're looking at in vitro systems more and more now. And one of the reasons that I'm really excited about this kind of work is because we've seen some echoes of what we've found to be bioaccumulative in humans who are exposed in similar ways. So we looked at the AFFF dosed mice, we saw what they were exposed to, and then we also went to El Paso County and looked at human serum from a community where their drinking water is impacted by a sulfonamide-based foam. So this is an area where PFOA and PFAS were found exceeding the then US EPA HAL, which has gone much farther down now, but it was 70 ppt. We knew that it was above that level. We didn't know when it started to be above that level. So we came in, I think, 2016 or 2017 is when we did the initial sampling. And our data looks kind of like this. So this is drinking water on the left from north, where the kind of source region where the AFFF releases probably were, down to south. So we're looking at 3 different water districts: Security, Widefield, and Fountain. And then we're also looking at the human serum from people who had lived for at least 4 years in those 3 water districts. And you can see reflected the same kind of thing. You have higher serum levels if you live in an area with higher drinking water levels. But what was really interesting to me about this was I also did high-res mass spec and suspect screening on these human serum samples. And these are the main compounds that we found that were not on our biomonitoring lists. So we have unsaturated PFAS, again, 85% detection, low concentrations, but frequency of detection is quite high. And then some of these oxygen substituted and ketone substituted perfluoroalkyl sulfonates. And these are all compounds that were also highlighted in our AFFF dosed mouse study. So it just goes to show that maybe these kinds of studies are a really good way to prioritize things that should be included on human biomonitoring lists. So to wrap up, I think what I'm hoping to convince you of is that we really need to test exposure relevant mixtures. It can be a difficult thing, especially to convince toxicologists to do, because they want to do one compound at a time. But some of these compounds we don't have analytical standards for. So we really don't have another way to prioritize or to figure out, what we should be concerned with other than using these kinds of mixtures. So we're doing these in vitro, in vivo, and now starting with in silico approaches to prioritize and hopefully inform biomonitoring using this kind of bioaccumulation directed prioritization. So in vivo models, in vitro models, informing PFAS biomonitoring. So I'd like to thank all of my collaborators and my funding sources, and I'm happy to take questions. Thank you again so much for having me.
One of the real concerns we fire, we've had a couple of episodes just in our general area. Is there somewhere down the future where we can find a substitute compound that can be used to replace what we currently have?
Yeah, that's a great question. Actually, As far as I know, right around now and in the next couple of years, we are phasing out the use of PFAS in firefighting foams. So there's all sorts of fluorine-free foams that are coming on the market, still looking for the best ones that perform as well. But more and more, we're understanding the risks. The days of using these foams with no PPE or spraying them all over during training exercises are definitely over. And when they are used, usually they would be the fluorotelmer C6 foams now, not the C8 sulfonamide foams, which are still PFAS, but they're shorter chain. But yeah, that's a huge movement right now to get to fluorine-free foams.
Is there long-term effect? Oh, thank you. I speak a lot, so I don't. Is there? Have you done any testing with any long-term effects from any of these fluoridated compounds?
Like health effects?
Yes.
So there's definitely people doing cancer research, and it's associated enough that the EPA now says that the MCL should be 0 because PFAS and PFOA are associated with cancer. So they're considered likely carcinogens, I believe, is what they've been classified as. So I think especially for our firefighters and first responders who use these foams a lot, it's a huge concern now that, they were using these occupationally for years and could be associated with some of the cancers that they get. They're exposed to a lot of chemicals. So it's always been a concern for them, but this is definitely a particular concern.
Other fish that have these compounds in them, but were there any land organisms that you could test that would be more comparable to maybe what humans are experiencing, or maybe different generations of humans in general, just to see how they're affected throughout age?
So that's a great question. I think there are a few studies, not a lot, that have come out looking at age differences in humans. There's a few from the CDC, looking at more the general population, children and adults, but those would be just limited targeted lists. And there's a couple from Stockholm University where they've looked at total organofluorine in different populations. So there is a little bit of that kind of data. I think generally people usually see higher concentrations overall in older people. But some of these other unidentified organofluorine kind of trends can be more confusing to look at. Yeah, we looked at the sharks because they're an apex predator, and they're also an understudied apex predator because they're elusive. So we have a fairly good amount of marine mammal data, and marine mammals have much higher concentrations than sharks do. So I think there's also some physiological differences. Sharks have different blood makeup and just weird bodies generally. There's a lot of reasons that could be. But yeah, I don't know what we would look at to be the most reflective of humans other than humans, because nothing really eats the same kind of stuff that we do.
Carrie, we have a question from our virtual audience. Have you attempted to identify and quantify pharmaceutical sources of fluorinated organics?
I really want to. We're thinking about it. There is one good paper that I saw that came out, I think, of Tom Webster's group, I want to say, where they did some demonstration that it seems like those pharmaceuticals are probably contributing, and it would make sense that they would be contributing. If people are taking fluorinated compounds, then of course that's going to show up in their blood. So I think I would like to see, to the best of our ability, us start adding them to our target lists.
Thank you for your presentation. 2 questions. So because of the sulfonate group or the carboxylic acid, activated carbon doesn't remove these compounds from drinking waters? Or is not that effective?
It does if they have long enough perfluorinated chains. So if they're
Sure.
hydrophobic enough, and if you replace them well enough, often enough, You can remove some of them, but for those really short chains, it's not going to be the most effective way to get rid of them. And then there's also the question of what do you do with that filter? These things there's no way you can't incinerate it. You'll just make shorter chains. You can't.
Right.
So what do you do with the filter?
Good point. The other question I have is, and maybe I missed this. So the type of ionization you're using for the mass spec, was that electron impact?
Electrospray ionization,
Okay.
negative, yeah.
Thank you.
Any other questions?
Yeah, I have one. So when you were doing the shark's blood, did you consider testing the water or perhaps even the soil to compare it to the blood in the sharks?
So we didn't do that with this study. We're actually working on a really cool study that we're starting now where we're going to do something like that with fish, mostly freshwater fish, which I think could be really interesting. With the sharks, I think it would have been difficult because they're so mobile that you really don't know, like they migrate down the coast every year. And they eat different things as they do that. So it'd be really hard to know what water would be representative.
Hello, because structurally these molecules look like the type of thing that would insert themselves into a cell wall. Do you have any assays planned or that you've seen that look at different types of tissue and see if there's trends in the variety of PFAS that are captured by them?
Yeah, that's extremely exciting to me. That's kind of where we're trying to go with some of this, because when you have a complex mixture and an in vivo model, you have way too many things going on when we have a living thing and an AFFF. So we're starting to look at some equilibrium dialysis methods and some other methods where maybe we can pull apart specific reservoirs like phospholipids and serum albumin and other transporter proteins that have an affinity and start to understand the structural aspects that make, some compounds have a higher affinity than others.
Any more questions? One more.
Oh, thanks, Michael. What do these fluorinated hydrocarbons do in the body? What's the mechanism that they're causing all these problems? Do they break down, or is it just they're there and they're near it like a splinter in your hand?
They don't break down unless they're precursors. So the precursors do break down to form perfluoroalkyl acids in the body. They accumulate in the liver and the kidneys to a high extent, and they're associated with dyslipidemia, some different kinds of cancer. Unfortunately, a lot of the mechanisms aren't super well described. We know that they seem to bind to PPAR gamma and some other receptors. They seem to activate some of the endocrine disruption pathways. So there's a lot, honestly, that they do. And a lot of that research is still, really underway, especially when you have this many compounds to study. It's really difficult to really fully describe the mechanisms, but there's enough, animal toxicity studies and in vitro studies to show that there are impacts.